Hello everyone. Welcome to today's live broadcast, Diagnostic Development and Laboratory Response to Infectious Disease Outbreaks. I am Christina Jewell of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational webinar seminar presented and sponsored by LabRoots. For more information, please visit www.labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Paula Revell. Paula A. Revell, PhD, DABMM, obtained her bachelor's degree at Washington University in St. Louis and her PhD from the Washington University School of Medicine, where she studied the bacterial pathogenesis of Yersinia enterocolitica in Dr. Virginia Miller's laboratory. Dr. Revell's postdoctoral studies included work on tumor immunology in Dr. Timothy Lay's laboratory, followed by a fellowship in medical and public health laboratory microbiology at Washington University Barnes Jewish Hospital and St. Louis Children's Hospital Department of Pathology and Immunology under the direction of Drs. Mike Dunn and Greg Storch. Dr. Revell is currently an assistant professor of pathology and pediatrics at Baylor College of Medicine and clinical liaison in the medical microbiology laboratories at Texas Children's Hospital. Her former positions include assistant professor at University of Texas Southwestern School of Medicine, Department of Pediatric and Pathology, Director of Medical Microbiology at Children's Medical Center of Dallas, and Director of Medical Microbiology Laboratories at Texas Children's Hospital. Dr. Revell has a strong interest in pediatric microbiology with a focus on molecular diagnostics as well as an interest in the evolution of gram-negative antimicrobial resistance. I will now turn it over to Dr. Paula Revell for her presentation. Dr. Revell?
Thank you for that introduction, and thank you to LabRoots for inviting me to give this talk today. So as Christina pointed out, the talk today is going to focus on diagnostic development and the lab's response to infectious disease outbreaks. So through the lens of my experience at Texas Children's Hospital, I'm going to walk you through some specific outbreaks that we have experienced and dealt with here at Texas Children's, as well as we'll go through some historic outbreaks for interest's sake. At the end of the talk, you should be able to define what an outbreak is, as well as discuss the specific role of the clinical or medical microbiology labs in the outbreak scenario. And then finally, you should be able to really contrast the steps that are required and involved in the validation of any assay that you may develop in response to an outbreak, and contrast specifically those assays that would be FDA cleared or approved versus those that would be laboratory developed. So we'll go ahead and get started. So what is an outbreak? It sounds, it's not just a title of a movie or something like that. There are a lot of different definitions that you can think of. Merriam-Webster describes it as a sudden or violent increase in activity, or an increase in the incidence of an infectious disease, or an increase in the numbers of harmful organisms, which like an outbreak of locusts or something like this. So from our perspective, we obviously focus on the disease. So the CDC, and this comes from specifically the FoodNet or PulseNet resource, which actually is involved in the investigation of any foodborne illness that may be associated with an outbreak in the United States. Their definition is that when 2 or more people get the same illness from the same contaminated food or drink, the event is defined as a foodborne outbreak. The World Health Organization tends to have a more global perspective, as you would expect, and their definitions are twofold. The first is generally an occurrence of cases of disease in excess of what you would normally see in a defined community, geographical area, or perhaps season, so point in time kind of thing. They also make an exception for certain communicable diseases where a single case that may have been long absent from the population or caused by an agent that has not been previously recognized in the community or area as an outbreak. So an example of that would be if we in Houston had a case of diphtheria, one case alone would be enough to constitute an outbreak for us. So outbreaks occur in space and time like anything else, and we define them based on the expanse of the region or the time that they cover. So in terms of location, a small or a local outbreak is what most of us probably see and are most familiar with on a hospital level. A local outbreak would be an example of an outbreak in your PICU or your NICU, or for those of you in the adult centers, your MICU or SICU. Another great example of a local outbreak would be a church picnic where everybody ate the same bad potato salad kind of thing. When we start to think of it in terms of a more public health perspective, there are also statewide or regional outbreaks, which by definition means it covers multiple counties within a state, and for some regional outbreaks can actually encompass neighboring states, but it doesn't quite span the nation. And once it does span the nation with multiple states across the country, we consider that a multi-state outbreak. And then finally, an international outbreak, sort of self-explanatory, outbreaks that basically cover the globe. A great example of a current international outbreak would be the pandemic that we're seeing with CTXM ESBL producing E. coli. So there's a specific urinary tract, community-associated urinary tract E. coli that's a very, very common occurrence across the globe, and this CTXM ESBL gene confers a high level of resistance. So we have patients from all over coming in from the community with the UTI that you historically could treat with Bactrim that now requires IV antibiotics. So it's not a specific disease in that case as it is the resistance phenotype that makes that an outbreak. So in time, outbreaks, unfortunately, are often defined retrospectively in terms of either the outbreak is over before we can officially define it, or it's been ongoing for a long time. And that's simply because sometimes they can be difficult to detect. Not always, but sometimes. And they can span very short periods of time or extended periods of time. The challenge is really to detect the outbreak in a timely manner in such that we can actually take action to intervene and hopefully effectively stop the outbreak or at least shorten it through our intervention. So we'll walk through a couple of historic outbreaks and then we'll get into more contemporary time. So the first outbreak that I think always deserves mention from an infectious disease perspective is the Black Death, or the plague, essentially. And this outbreak was caused by a gram-negative organism called Yersinia pestis. This is gram-negative bacteria, and it was carried by little fleas that infect rats, and it caused bubonic or pneumonic plague. During the 1300s and medieval times, essentially, there are estimates that as high as 75 million people died when the Earth's population was only 450 million total. So this was hugely devastating. There's nothing that could even come close to it with what we see in current time. Half of Europe died, for example, in 4 years. So this is the the granddaddy of all outbreaks, if you will. And we're very fortunate to not have to deal with anything quite this significant today. Today, probably the most common and the most significant outbreak that we deal with every year is influenza. So influenza causes yearly epidemics that are caused because their genetic composition changes just subtly. We call it antigenic drift. So the viral genome actually subtly changes with time due to the nature of its replication. And every year the virus can change enough that it can kind of break through and cause another epidemic. That's why we have to constantly be diligent in terms of observing which virus is circulating and updating our vaccine. Pandemics actually happen when the virus does what we call a shift. So this is the virus itself has a segmented genome, and when those segments reassort, so commonly we see swine, avian, and human. Any kind of shuffling, that's typically when we see a pandemic. During a typical epidemic or outbreak of influenza during a typical season, we see anywhere from 150 to about 250 deaths yearly. So this is not trivial, even today. Most severe cases are in the very, very young or in the very old, in the elderly. And people will be angry because this is not my definition, but they say elderly is over 65. I didn't come up with that number. But essentially, greater than 90% of our fatalities occur in older patients during typical epidemic outbreaks. Pandemics are different. Pandemics, the best, most dramatic, if you will, example of influenza pandemic occurred during the 1918 Spanish flu pandemic. And essentially a third of the world's population is estimated to have been infected with this virus. And the unfortunate thing is that the virus had a death rate of 2.5%, and thus there was an estimated death of around 50 to 100 million cases. In fact, you may know this, but the pandemic Spanish flu actually caused more death than World War I. So it was hugely impacting, as many of these can be. Our recent memory will bring to mind 2009. At that point in time, we called it swine flu. It's now a novel H1N1. But the 2009 pandemic actually resulted in 120 to 200,000 deaths. But the significant thing, and this is the case with all influenza pandemics, that differentiates them, aside from just the overall number of those that become infected, because it's a naive background, is that most of these deaths during the 2009 pandemic occurred in patients that were less than 65. So these were normal, healthy adults, many of whom died as a result of contracting this virus. So I'm going to talk about a lot of other outbreaks today, but I'd like to just remind everyone that influenza still today is the leading infectious cause of death among children and young adults in the U.S. And I always give a talk with at least one point saying you should get your flu shot. So that's my plug for today. I have no association with influenza shot development. I just think it's good for your health, good for the health of your community, good for the health of our children. So another current outbreak is actually a bacterial outbreak. And this is something that was picked up on by the CDC due to our FoodNet, our PulseNet, which is this infrastructure we have within the U.S. that the CDC, through the various state labs, tracks foodborne outbreaks. So on June 1st of 2016, the CDC announced that they were doing a multi-state investigation due to outbreaks of Shiga toxin-producing E. coli, in this case, 0121. So the most famous Shiga toxin-producing E. coli is the 0157, and if you all think back to the Jack-in-the-Box outbreak, where we actually saw fatalities associated with it when the undercooked meat. This is another example of a Shiga toxin-producing E. coli. And as of July 25th, there have been 46 people that have been infected across 21 different states. So this is a national outbreak. Fortunately, there have been no deaths, but this outbreak was, through all the investigations that have been done, linked to General Mills flour. And so that may be why you're Kroger or Schnucks or wherever your grocery store is may have sent you a note or called you to say, hey, if you bought this 2-pound bag of flour, please return it. It's not healthy. So that's an example of another current outbreak that we're dealing with. So this slide just demonstrates that every day there's something new. All you have to do is just turn on the news. We have Ebola, we have yellow fever, we have MERS, which is the Middle Eastern respiratory virus, which is a coronavirus similar to SARS, which is, on the cover of Newsweek you see here, Bluebell ice cream recall due to listeria, chikungunya, you name it, Zika, we've got it. So we always have to be alert to these outbreaks. So what's the role of the clinical lab? What's the role, my role as a lab director or clinical consultant? And that is twofold. So essentially, our first and always biggest priority as a clinical lab is the patient perspective. Our job is to identify a specific infectious agent and provide data that would allow the physician to appropriately treat. So if it's a bacterial infection, that's antimicrobial data through susceptibilities. If it's viral, it may simply be just identification of the organism so that they know it's influenza, let's prescribe Tamiflu. From a public health perspective, it's more complicated, and we play essentially roles as sentinel centers. So clinical labs are like the canaries in the coal mine, if you will. We are the early detectors. So the first case of that Shiga toxin-producing E. coli 0121 was somebody's clinical lab. And so our role is to detect and identify and report. And we have a very well-established reporting structure through our local infection control, through our state and national public health, and then all the way up to the CDC. And finally, not all labs may have this capacity, but many do, and such as ours, and that is to assess relatedness of the organisms. So when you think about it, even if this doesn't go beyond just your own individual hospital, it's important to know whether these bugs that you might be detecting are just random and sporadic, or are they truly related and thus, by definition, constitute an outbreak. So what do we look for? We look for some specific pathogens, so listeria, brucella, shigella, salmonella, anthrax, measles, but those are not the only thing that constitute a potential outbreak. We also look for any increase in what we might refer to as a normal human pathogen, so pneumococcus or Staph aureus. If we see an increase in numbers, that may be enough to alert us to something going on. Also, it's important to note that sorry, I'm in a conference room that just shut the lights off on, I wasn't moving enough. We identify any uncommon resistance patterns or potentially new resistance patterns, and that's one of the things I alluded to earlier in terms of the CTXMs, ESBLs. Carbapenem-resistant Enterobacteriaceae are a great example of that. This is something that if we see this type of a bug that we don't normally see in our population, we need to identify and communicate. And then also, obviously, the identification of any new or uncommon pathogen. So who do we help when we do this? Our priority is always to the individual patient, but it goes beyond that. And this really is a public health issue. So we help the health of the hospital, the local community, the national community, and the global community by all playing a part in terms of this team of investigators who can help detect and potentially stop these outbreaks. So how do we identify these pathogens in the clinical lab? Frankly, we don't have any special magic toolkits. We just use our traditional methods. So for bacterial identification, we grow bacteria, we grow a virus, and we culture them and then identify them using traditional methods. There's also an evolving field of molecular detection that allows us to detect things, in particular in the realm of virology, faster than we could with many of our old traditional methods. And then in some instances, we may never be able to culture these bugs, and so everything is defined and detected simply by serologic detection, which is looking at the host and how did the host respond to this virus to determine whether or not, or bacteria to determine whether or not they've been infected. So this is an example of one of our local outbreaks that we dealt with. And essentially in mid-April, we had an MRSA identified from a routine blood culture of one of our patients in the NICU. It's not common, but one sporadic case like that does not make an outbreak, and it doesn't even raise too many red flags when you have very large NICUs like we do. This type of infection is unfortunate, but can happen. But in early May, we had one of our bench techs, and this emphasizes that this is not just a one-man game. It's really a team game, and many of these investigations that you'll hear about today, the first person to be that canary in the coal mine was just a bench tech who happened to be paying close attention and said, hey, you know what? I feel like I've noticed more MRSA isolates lately. And so that word leads to myself as a director to do a little bit deeper investigation and notify our infection control. And indeed, we did assess that we had an increase in our number of MRSA isolates. So after that, the laboratory, we went ahead and did an assessment of potential relatedness. And by that I mean you can have just a random increase in numbers that are true, and unrelated. So we may have an increase in MRSA infections, but may have nothing to do with each other. So we went ahead and did that assessment, and indeed we did have a clonal strain that was causing these infections. And so at that point in time, infection control really initiates a formal investigation to look for the source and to implement the bundles and practices that we need to actually stop the outbreak. So this is what a picture of what I'm describing as our assessment of relatedness. And essentially, I'm going to try my best to draw an arrow here. This group here, so 13 to 1, if you look, all of those isolates actually have the same, what I would call, banding pattern. And by that, I mean, if you look at each of, these little bands, they are unique. They are unique for 18, 17, 16, 15, and 14. But if you look at those from 13 through 1, essentially they all have the same banding pattern. And you can tell by down below here, you may not, I'm not sure how well this projects, but this is the percent similarity. And so essentially, all of those 13 through 1 hover right around here at around almost 98% similarity. So that really does indicate that we're looking at the same bug. So what's our biggest challenge in the lab? And I'd say that the biggest challenge is how do you detect something when you don't know what it is? So we always need to be alert to these outbreaks, but we may not even know what it is that we're looking for. And I apologize for fellow Iowans. I'm a native Iowan, and so I can make fun of Iowa in this slide. And this is really what I have as my image for what started that influenza pandemic of 2009, the mixing of the human and the swine. So I'm going to walk you through now what our experience was in terms of really something that no one had ever seen before, and that is a completely novel and new influenza virus, and how our lab was actually able to respond and react appropriately. So this is a timeline that initiates back in, excuse me, early April of 2009. There's a 39-year-old woman who dies in Mexico of what at that point in time was called an atypical pneumonia. Then on the 20th, we hear reports of over 800 cases of this mysterious atypical pneumonia in Mexico City. So clearly there's something, there is an outbreak in Mexico City. What is it? Nobody knows at that point in time. And fortunately or unfortunately, however you want to look at it, for us, we then saw in, excuse me, mid to late April, there were 2 cases of pediatric, what at that point in time was called swine influenza in children in Southern California. So on April 27th, the WHO raised its pandemic alert level to 4, which means that there is human-to-human transmission, and it's enough to sustain community-level outbreaks. And then, unfortunately, on that same day in Houston, in our emergency department, we had the first reported death in a child who had traveled to Houston from Mexico who had H1N1. So this was sort of the opening of the floodgates for us. So our diagnostic capacity at that date and time was that we had the rapid flu, which you all know, it's just the little thing where you go into your doctor's office, they stick a swab in your nose and tell you yes or no, you have influenza. Okay, we didn't have any idea whether or not the existing rapid influenza tests could detect this novel virus. If it could, how well could it detect it? And if it told us it was flu A, we couldn't subtype. So we didn't know enough about whether or not we really were seeing that novel virus. We had culture, but that was slow, and we couldn't subtype there either. And there were no molecular detection assays. So you might ask, well, at that point in time, what were the actual number of commercially available molecular assays to detect influenza? The answer to that was 0. So no matter how many years pediatricians had been saying to various companies, we really would like an influenza molecular test, you know, that just wasn't what people were interested in before the pandemic. Things are different now, but at that point in time, there were none. So one thing that is important to know about Houston is every year we worry about hurricanes in Houston. And I put this picture here up because at TCH still today, we have the legacy of Carla, and that is everybody has to have in their office, if you're essential hospital personnel, you have what's called a Carla bag. So if the hurricane hits and Houston shuts down, you stay and the hospital doesn't. So we can continue to run in a crisis situation. And that's one of the things that Texas Children's does very well is crisis management. When I arrived in Texas, I arrived actually on April 9th of that year, 2009. And I got called into the command center on that day on April 27th, and they had one question for me, and they said, can we test for this virus? So my answer to them was, well, we have a couple of options, and they were that number 1 and number 2 were that we could potentially do what we call a laboratory-developed assay to detect all influenza A using molecular means, and then to potentially subtype, or we could just wait, because at that point in time, we knew that the CDC had an assay, but they had not released the sequences. Even the state labs, nobody else could do testing except the CDC. So, knowing that, fortunate for us, the 2 cases in California that had been reported in the children in California, they had sequenced both of those viruses, the complete genome, and deposited it into GenBank. And that actually is a gold mine for laboratories with the capacity to do diagnostic development, because it allowed us the access to the information, what we call in silico, to know what we needed to target in terms of a molecular assay. So at that point in time, rather than wait, we decided to go forward with our development of a homebrew assay. So what do you have to do something like that, to bring an assay like that up? Well, assay development, assay optimization, and validation. Sounds easy, 1, 2, 3, but it's not quite as straightforward as it sounds. And it's important to remember that there are a variety of regulatory agencies who are always looking over our shoulder as laboratorians when we do this kind of test development. So first and foremost, there's the FDA. And the Food and Drug Administration, what they do is they regulate the development of tests primarily by manufacturers who are then going to distribute the test. The College of American Pathologists is more pertinent to those individual labs who may develop a test and then only use it within their own patient population. And then obviously all labs are have to abide by the Clinical Laboratory Improvement Amendments, or CLIA. So in terms of the FDA designations and what the FDA does, for device manufacturers, and I will be using the term device when I'm describing an assay, a detection assay, and that's simply because as far as the FDA is concerned, all laboratory tests are actual medical devices. So there are 2 different types of clearance or designations that you can get through the FDA. The one is a 510K, referred to as FDA cleared, and the second is referred to as FDA approved. So any manufacturer who makes a kit that can actually sell it to a clinical lab and that you can then test and use in your lab, it's either FDA cleared or it's FDA approved. If it's not, it receives a designation of what you would hear as research use only. And typically, if it is an RUO, or research use only, you cannot report it clinically unless you've done a complete internal validation, which we'll talk about in a bit. But the big difference between an FDA-cleared test and an FDA-approved test is simply the level of data that is required for that designation. So any FDA-approved assay actually has gone through extensive clinical trials, and there is data from real patients that can say this is how this assay impacts patient care. So great examples using outcome data are HIV quantitative, HIV qualitative. These are FDA-approved assays. Most of the assays that you're familiar with, so whether it's influenza or any of the other viral respiratory detection kits that are out there, they're called FDA cleared, and that simply means that the laboratory performance has been established. It doesn't mean that it's gone through extensive clinical trials to show that the patient outcome is better when this test is used. So what about all the other tests? A lot of tests done are not things that we get commercially from from companies that have been cleared or approved by the FDA. They're what we call laboratory developed tests, or LDTs. So these assays include anything that's developed internal to your own lab through research and development, but importantly also anything that is a modification of an existing FDA cleared or approved test. So those modifications can be what seems trivial to you, but it's enough to mean that this is now a laboratory developed test and the validation process is extensively different. So if you change the specimen type, if you change the collection device, the assay platform, the extraction platform, if you make any modification to that test, it becomes a laboratory developed test. So, validation. What do you do? Well, for an unmodified FDA cleared or approved test, we have to establish accuracy, reproducibility, reportable range, reference range. And accuracy in this instance, this is a misnomer. I don't know how to, let's see if I can make a big X through this. That should be gone, so ignore that right there. The accuracy in this case simply means agreement. So you need to show that if the company says if the test says this, and this is positive in the company's hands, then you need to be able to come up with the same result. Reproducibility simply means it's precision. So if Tech A does it and Tech B does it and they both do it 5 times, it's the same answer all 5 times. Reportable range means that when you're doing this validation, you should really evaluate things at both the high and low levels. And the reference range means that you really should be documenting that you're not detecting things below the what we call the limit of detection. So all of that has to be done anytime you validate any test in-house. But if you are developing your own laboratory developed test or modifying any FDA test, you also have to establish the analytical sensitivity and specificity. So that means how sensitive is that test in terms of its ability to pick up, and then how specific is it? Is it cross-reacting? If you're looking for influenza A and you're calling it influenza A, you need to make sure that it's not going to be positive if it's rhinovirus, if it's flu B, whatever the case may be. So think of specificity as kind of cross-reactivity, and sensitivity is how sensitive is this tool to be able to detect this test. So I'll emphasize again, the biggest difference there in the validation of an FDA cleared test versus validation of a lab developed test is that additional analytical sensitivity and specificity. So this is just some information from sources that can help you through your validation process. The Cumatech 31A, it's titled The Verification and Validation of Procedures in the Clinical Microbiology Laboratory. They actually have sort of a nice guideline in terms of what you should do for validation of an assay. And if you have a CAP inspector come through and they want to look at your validation, chances are they're going to reference this Cumatech as what you should have done as the minimum validation requirements. They recommend a minimum of positive specimens, 100 negative specimens for laboratory developed tests. So that means, sorry, the lights just went off again. That means if you are doing a lab developed test, you can't just test 10 specimens and say that's good. the FDA cleared assays and the approved assays, all you have to do is show agreement. So a minimum of 20 specimens, and you should have roughly 10 positive and 10 negative, but you need to show agreement with another validated platform with those numbers. So back to influenza, we had decided to do a complete laboratory developed test. And so what that means is we started out with, and this is and it really is not projecting well here, but over here you can see all of these are viruses, a variety of H3N2, H1N1 across the years, but these are all influenza A viruses. And this here is the genomic sequence as was published on GenBank for the matrix gene of all of these different viruses. And so what you can see here highlighted in yellow was an area of complete similarity. That means every single one of those little letters, the A, T, C's, and G's, they're all exactly the same within this region. Down here, there are a few differences, like it can be an A or a T. And over here, likewise, there are a couple of A's versus T's. So these regions, though, as you can see, regardless, across the years and across the subtype, the matrix gene is highly conserved. So it's a great target to pick if you're going to look for all influenza A. And that's what we did. This is where we chose our forward primer. This is where we chose our reverse primer. And this is where we chose our probe. So we went forward based on that with our validation. And when we did the validation, it was kind of an ongoing validation because what you have to have is a gold standard. So obviously there were no existing FDA cleared assays at that time, so our gold standard was the molecular assay that the CDC was performing. We couldn't do it ourselves, we didn't have the primers and probes, but at that point in time we were sending every single positive that we had to the CDC. And at the beginning, they were turning them around pretty quickly. As the numbers increased and as the awareness across the country became greater, the time to result from the CDC became longer and longer, but it allowed us to go through our validation process nonetheless. And what you see here is that we have our assay here. This is our homebrew RT-PCR, positive and negative, and the CDC assay, positive and negative. And so we have complete agreement here and 100% specificity and sensitivity. This was early in the validation, so you'll note that we weren't quite to the 50 positives yet, but it's just a snapshot in time. When we actually compared to culture, our culture to our PCR, the results were actually quite shocking, and that's what this slide is showing here, and that is that we had 292 culture negative but PCR positive specimens. That means that the sensitivity for our culture was just below 60%. So had we decided to only use culture as an identification method, we would have missed almost 50% of our positive patients. So this is what the results came out for our EIA, or the rapid flu, as you remember. And as you can see, EIA positive, negative, EIA positive, negative, real-time PCR positive or negative. So again, this is the important number. 378 flu negative rapid tests were indeed positive by the PCR. That's a sensitivity of less than 50%. That's not even as good as a good clinician. So it's very good that we didn't depend on what at that point in time was our existing tool to detect this virus. So what did we learn through this whole process? We learned that, the real-time assay is more sensitive than the rapid or the culture. And we also realized that we could, using our own staff and our own processes, really turn these assays around, or turn the tests around in time to impact patient care. So this is back to our timeline. The day we lost that patient, ordered our primers and probes and started our validation. And it was 3 days later that the CDC finally released their primers and probes, but we were all well underway in terms of testing and validating our assay, so we decided to go forward with our homebrew assay. On 5/1, our preliminary data looked good. We had the 50 positives that we needed. It just shows the significant numbers of patients that we were seeing through this time. And by 5/21, we were testing any patient who presented with respiratory illness to our hospital with this real-time assay. Importantly, and this is a very important note, as of June 1st, labs that were using the CDC assay started running into massive shortages of reagents. So they couldn't get the supplies that they needed to be able to do the test. And that was a real eye-opener for us. We hadn't anticipated that one. But by the first week in June, we tested over 1,000 specimens, and roughly 300 of them were swine positive. And we continued throughout the next essentially 2 years with this testing. So we learned that our standard methods weren't going to work, and we really needed the in-house development. We also learned, importantly, and these are two very important points here, and the one is that the commonly available agents in these situations where you have a massive outbreak, where you have this big surge in infection, common reagents that everybody else are using can become shortage, or the limiting factor. Infrastructure, specimen delivery, all these things were critical. And we learned, we had doctors bringing Sam's freezer bags, holding patient specimens, going through with patient identifiers. It was a zoo. We were seeing about 300 patients an hour through our ED just because of influenza. So we also learned that automation is critical. These high throughput platforms really can save us in these types of outbreak situations. And then also, and this is an important lesson, and it's not a negative about the CDC and our public health infrastructure at all, but we did learn that the CDC and our state labs cannot give us results in time to impact patient care or infection control practices. And when you think about it, that's not their job. Their job is to maintain the public health infrastructure of the country, not to respond for that patient, the initial patient. That's our job. That's the clinical lab's job. And that's why our ability to develop and implement these assays in-house and not exclusively depend on our public health infrastructure is critical. So we finished the pandemic. That was going to be the end of the story. We weren't going to worry about it anymore. But then we had a few years where we didn't think too much, just the little local salmonella outbreaks, maybe listeria here and there with our Bluebell ice cream that everybody loves in Texas. But we didn't quite have anything quite as scary as the pandemic until we received a call from our colleagues in Dallas with Ebola virus. So, Ebola seemed like an exotic disease, very far away in West Africa. And to be honest with you, as a lab director, it really wasn't high on my list of things that I was even thinking about until we got this call. And that is the index patient developed symptoms in late September of 2014, about 4 days after arriving to the US from West Africa. And when he started feeling ill, he presented to the community hospital, Dallas Health Presby, on September 26th. He came back to the hospital and was admitted on the 28th, and testing done confirmed that he did indeed have Ebola virus disease, and he died on October 8th. So this was a huge eye-opener, because any of you who are geography buffs should know that Dallas is just up 45. It's about 3.5 hours north of us, and so if it can happen there, it can happen to us very easily. So post index case, we saw 2 additional cases, and the first was one of the nurses who actually cared for the index patient. She was self-monitoring for fever, so she presented to the hospital with low-grade fever, was diagnosed with Ebola, and then recovered and released from the NIH Critical Care Center on October 24th. And the third case was also a healthcare provider who cared for the index case. Again, self-monitored, everything worked like clockwork, went to Emory, was admitted, recovered. But it was a real eye-opener for us in terms of we need to be able to respond not just to outbreaks in terms of detection and surge capacity, but some of these viruses that are extremely infectious and extremely dangerous, we also need to have additional capacity there. And so our response to this was that we opened the first pediatric special isolation unit in the country. And what this is here, you can see in this picture, this room here is actually the patient room. And so there are no beds here at this point in time because this is right prior to opening. But what you can see is this little anteroom here has many of the monitors and computers. So when you are going to enter a patient's room and you're really just looking and monitoring and looking at the monitors, you're not going to lay hands on the patient, not going to, actually listen to their heart, this kind of thing. Having all of these separated out to this relative to this room is critical, and this room is all under negative pressure. And importantly, the observation windows allow people to visualize but not be in the room. And then we have additional development for the siblings and families in terms of playrooms and things like this that are removed from but nearby this isolation unit. And there is a BSL-3 laboratory, which means that we have the capacity to work with pathogens. You don't want to be growing Ebola, obviously, it's a BSL-4, but in terms of detection, working, whether it's TB, whatever the case may be, we do have a specialized dedicated lab immediately adjacent to these rooms. So we also decided we had to develop or implement, I should say, an Ebola virus assay because we couldn't very well, knowing how easy it is for somebody to walk off an airplane and present to your center, not have the ability to do this. So there were two commercially available kits at that time. One was what we call high complexity, which means it required multiple touches, highly trained molecular tech to run the assay. The other was what we call sample to answer. So that's these kits that are these enclosed little pouches or cartridges, whichever the case may be, where you simply insert your specimen, put it on the machine, and it gives you the answer. So, excuse me, we opted to go with the sample to answer. And we validated using contrived or spiked specimens. And fortunately, we never had to use the assay. We did have a handful of patients that presented and actually were flown from DC and various areas in the country to the special isolation unit from West Africa based on symptoms. But fortunately, all of the patients simply had flu, so that was good. Now, our next and current outbreak that we're dealing with is Zika. And so what is Zika? It's a virus that actually arrived in the Americas, probably to Brazil from French Polynesia, between May and December of 2013. And in August of 2014, physicians in northeastern Brazil began to investigate an outbreak of what really symptomatically looked a lot like dengue. So then in May, researchers determined using RT-PCR that that illness was actually caused by the Zika virus. And it wasn't until October of 2015 that an increase in the number of reported cases of microcephaly was reported to the Brazil Ministry of Health. And by January of 2016, there were almost 4,000 suspected microcephaly cases associated with Zika. In the early days, we obviously didn't know whether that was just an association, true and unrelated, but we now do know that the Zika virus actually does cause these significant birth defects. So what is Zika? It's a flavivirus. It's closely related to dengue. It's closely related to West Nile, yellow fever, and these viruses are all spread by mosquitoes. They're spread by the Aedes mosquito, which we have in Houston, which we have in Florida, which we have in many parts of the country. You can also see transmission from mother to child through maternal-fetal transmission and through sexual contact. The symptoms are very nonspecific and really don't negatively impact the individual unless the patient is pregnant. You can see these severe birth defects such as microcephaly, which was our primary concern as a pediatric center. We do now have a handful of cases of what we call local or autochthonous transmission, and that means that the individuals with Zika did not travel to Central America or Brazil or wherever and come back with the virus. It was not travel associated. They actually acquired the virus in South Florida. So this is just a map, and what you can see is this represents the dark blue, so Texas, California, Florida, Georgia. These are all states where we've had more than 50 positive Zika cases. However, all of these solid colored states are travel associated only, okay? And then the lighter the color, the fewer the cases, okay? Down here in Florida is the one exception where it's dark blue, they've had more than 50 cases, and they have this hatched line here. That indicates that there actually has been local spread. And so this obviously is critical in terms of our thinking of the ability to detect this virus, simply because we have not just a large standing pediatric center, but we also have a large standing women's hospital where we deliver hundreds and thousands of babies, and we have a lot of very nervous moms. We are in an area where we have the mosquito. We are in an area where we have a tremendous amount of travel back and forth to Mexico, South Central America. So we really decided that we needed to be on the forefront of this ahead of time rather than be caught up on the back end. So we decided to go ahead and develop an assay to detect Zika. So with collaboration with Methodist Hospital and the L.E. Simmons Viral Foundation, we were able to develop the assay, the first available in the U.S. to detect this virus. And how did we do that? We actually started testing on February 22nd, and we test using real-time PCR, serum, amniotic fluid, urine, and CSF. We test Monday through Friday. And per the CDC, the serum is what we test within the first 7 days of symptom onset. The amniotic fluid we use to diagnose intrauterine infection. And we do offer testing. We have a special Zika clinic for women who have clinical illness within 2 weeks of travel. And we know that urine can be positive with up to 30 days post-infection. So we actually went with an assay in that case that was commercially available in Europe, but there were no FDA-approved assays. And we did a similar type of validation that we saw with Ebola, where we use what we call contrived samples. So we spiked specimens with virus that we'd received from one of our colleagues who is a researcher in Colombia, and that allowed us to validate the assay and then go live. And I would emphasize, Zika is a bad player if you're a pregnant mom, and I've been a pregnant mom before, and I know there are a lot of things out there to stress you out. So just remember, the best defense against a virus like this is to simply wear mosquito repellent. So that's all I have today, and if anyone has any questions, you can type them into the Q&A. Otherwise, thank you very much for your time.
Thank you for that informative presentation, Dr. Revell. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will try to answer as many questions as we can. As the questions come in, and Dr. Revell, did you have any last comments before we answer any questions?
Sorry, I was going to type it in. No, I mean, I would just emphasize that every lab has to remember that we're all part of this system, and it's our role as the Sentinel Centers to really play a key part in the health of our public health infrastructure. I guess one final plug that I would have is, even if your lab doesn't do the types of assay development or homebrew lab developed tests that our lab does, I would like to emphasize the importance of reporting and sending isolates to the CDC through your state health lab, because that's really what allows us to go from the isolated hospital type outbreak to knowing and ensuring the health of our regions and our countries is this global public health infrastructure that we have. And our global public health infrastructure is really only as good as those of us who contribute on the front end, on the front line. So I would just emphasize that. And that's it.
Thank you again, Dr. Revell. And it looks like we don't have time to answer any questions today, but if you did submit some, Dr. Revell can answer them via email. I want to remind you that today's webcast will be available for on-demand viewing through December 7th, 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.